Welcome to week 11 office hour, all about fermentation. Last week, week 10, we went through cellular respiration, and we said that the first step for the organisms is to break down the glucose, oxidize the glucose to pyruvate. At this time, ADP got phosphorylized. This was a substrate level phosphorylation. ATP, I put here small amount with the help of NAD plus, as it's got reduced to NADH. This is reduction, and the glucose got oxidized. There's no oxidation without reduction, as well as vice versa, no reduction without oxidation. The pyruvate, and last time we saw it in the presence of oxygen, it went through the Krebs cycle, and also it went through oxidative phosphorylation with the electron transport chain, as well as the chemiosmosis. And we learned that we had a ton of ATP with that process. Now, what happens if there's no oxygen and no electron transport chain, but the organism still would like to get some ATP production? So pyruvate without oxygen. Oh, I should have written O2. I will do that without oxygen. So based on what type of enzymes an organism's cytoplasm contains, because we are still in cytoplasm, how we were in when the glucose got oxidized to pyruvate. This was actually that process, was the glycolysis. Glycolysis, depending on what they have, it can lead into lactic acid as one of the end products. In order to form lactic acid, the pyruvate needs to get reduced. NADH is going to reduce this to the pyruvate to lactic acid, and NAD plus is going to be one of the end products. Why is that super important? Here, check it. This NAD plus is required for glycolysis. The point of the lactic acid fermentation is to fuel the glycolysis in the cytoplasm with NAD plus for the glucose to be oxidized to pyruvate, while the NAD plus is going to get reduced, picks up the energy, high energy electrons, as well as hydrogens from the glucose to pyruvate. That is really the whole purpose of the lactic acid fermentation. The other product, depending on, not the other way is the alcohol fermentation. Ethyl alcohol fermentation. The ethyl alcohol is 2-carbon molecule and the pyruvate is 3, so 1 CO2 will leave, as well as to reduce this pyruvate to ethyl alcohol, the NADH is going to get oxidized. So this is oxidation, this is oxidation, and at this point, this is reduction, and that is reduction too. So ethyl alcohol. Now, which one is going to happen in an organism depends on its enzyme, depends on the presence of the enzyme. If there's lactic acid fermentation, it is because the organisms, the cell contains only enzymes to reduce the pyruvate to lactic acid, or vice versa, or the other way, ethyl alcohol. I cannot produce ethyl alcohol via fermentation because I don't have enzyme for it. Yeast can do it, but yeast cannot do lactic acid fermentation because the yeast contains only enzymes that goes this direction. So this is what I circle around now, is the fermentation. And the point of the fermentation is producing NAD plus that would fuel the glycolysis in the cytoplasm. Here is also in the ethyl alcohol fermentation. This NAD plus is going to fuel the oxidation of glucose to pyruvate in the cytoplasm. And that's all, guys, for the fermentation. So before I just close, please remember, fermentation does not produce ATP. Fermentation is only fueling glycolysis that makes the ATP. Have a good one.